them learn on their own.
Is it possible that a human and a donkey reproduce, or is it
Well, yes,
like
you could take the genes of a human and splice them using CRISPR into the genes of any other animal, and the thing will most likely reproduce.
what the?
Oh,
So are you telling me there's a possibility that half human, half donkey could happen?
No, no, that's not the way that it would be. It would be small sections. There'd be tiny, tiny sections of DNA that you could switch out. Like if there, like for the DNA for the placenta is the same in all mammals that have a placenta. The platypus and a couple others are too basal. They're different. But you can take the placental genes from any human being, and using CRISPR replace them with the placental genes of any mammal that possesses placental genes, and it'll work just fine.
and it's the same size.
Yes, same size.
Oh wow. Okay.
Yeah,
So
it's just coding for a couple of different proteins
Yeah,
that
it's
allow the egg to bond to the wall of the uterus.
oh
And
wow.
that they did a study, I don't know, maybe 5 years ago, maybe more recently. I'd have to look it up. But they were finding all these chunks of viral DNA kind of in the human genome, right? That matched with DNA or RNA strands that viruses have. And they found one that codes for a particular protein that matches the protein coding for a virus. How the virus protein it produces on its sort of shell to allow it to bond onto its host. That coding for that protein is the exact same coding in humans to produce the protein that bonds the egg to the wall of the placenta. So the theory is that evolution from non-placental to placental mammals occurred because of a viral mutation.
And this is like all conserved within the like all the animals that produce placenta.
Yeah, this would be any placental mammal.
any mammal that has a placenta. You can using CRISPR switch out the placental genes. They're all the same.
So I used CRISPR one time, but I used deactivated Cas9 protein to essentially label like certain DNA to see how cells reorganize their genetic DNA when they're going through cell division and stuff like that. And I can say like 100%, like CRISPR is not as accurate as you guys think.
Well, I know it's not right now, but you can still do what I'm saying that you can do.
Maybe.
I think I can find the study.
Yeah, because when I used it, it was mostly off target. I mean,
How long ago was that? How long ago was it?
that was like 2021. So
So like 2 years. It has come a long way in the last few years, but it's still it's not that accurate. But all you got to do is account for the margin for error. It's only a few locales. It's not that big.
yeah, that's the problem. Is that the guide RNA is not that long, so it could target regions that it's not supposed to target, basically.
Well, sure, but you can do it in you can the placenta. That's why they chose the placental DNA because it's surrounded by a bunch of LTRs or LTLs.
Yeah, but the guide RNA that the CRISPR uses is like it's only 12 nucleotide long, right? So those two. I mean, if those two,
But see, you can do it far enough out so that all of that margin for error is in the LTLs or LTRs, long terminal repeats, LTRs. The entire margin for error for CRISPR can be accompanied in the LTRs for the placental DNA because it's an ERV.
an ERV.
Yes, it's an ERV. The placental DNA is ERV in all mammals.
What do you mean ERV? Like,
Endogenous retrovirus. It's
oh,
retrovirus DNA.
oh, okay, endogenous retrovirus. Okay.
Yeah, and so the way that endogenous retroviruses insert themselves into the DNA creates long terminal repeats at both ends, and the entire margin for error of CRISPR can be encompassed by those LTRs without ever touching the DNA for the placenta.
Yeah. So when are you like trying to talk about how like the how we insert the DNA that encodes for whatever CRISPR into the mammalian cell? Like, is that what you're talking about when you say?
Into any DNA strand doesn't matter, but I'm talking specifically about mammalian DNA. Yes.
So, yeah, I would imagine like you probably use like How exactly do you get through this the cell membrane? Is it like I forgot? I think we use like
We use
virus,
needles to get through the cell membrane.
huh?
We use needles to get through the cell, just like you do with in vitro egg fertilization. You use a
No,
needle.
no, cells are too small for that.
Well,
No,
okay, I don't know the exact method. It's in the paper. I didn't read the entire method because it's long and boring. But I read the abstract and the conclusion, and I read as much of the method as I could.
yeah, I forgot. I think
I could probably find the paper if you need me to. I mean, if it's this big a deal.
yeah,
But I've explained to you how they compensated for the margin for error that you're talking about without ever damaging the DNA. And you can do that because that's the same in all mammals because there's LTRs at each end of the placental DNA in all mammals.
yeah, but like that's what I was talking about. Maybe I wasn't clear enough, but like how CRISPR works is that it has a guide RNA, right? And it also has proteins called a Cas9 protein that essentially cuts the DNA. It breaks the
Yes.
DNA.
Yes, I know.
So how it targets the DNA is essentially using those guide RNA that it has, and it uses it as a complementary, and it binds to the DNA, and then it makes
Yes.
a decision, right? So those, like from what I remember, those are, I mean, that RNA is only 12 nucleotides long, so it could essentially, I mean, it's only like four, what is it, four nucleotides. So if we have only
So
12,
LTRs are hundreds of nucleotides long. You can drop it anywhere in the LTR and it encompasses the margin for error without touching the placental DNA. I've already explained this.
yeah, but okay, so where would it cut then? Like
Anywhere in the LTR.
it's gonna cut in the LTR, okay?
Anywhere in the LTR because it's the same in all mammals. There's huge amounts of LTRs at the outside on both ends of the placental DNA in all mammals that have placental DNA.